Okay, thank you very much for the introduction, Michael. Just as a bit of a background, I'm a research scientist from Curtin University working with the Centre for Mine Site Restoration. Just bear with me for a second while I get my presentation up and going. Okay, so that's kind of working. Yeah, look, sorry about that. You always get these hiccups with these types of things. So basically, I'm going to be talking about a species called Nuytsia floribunda, which is a species that's found in the southwest of Western Australia. The reason I'm talking about this really is because it's been affected by sand mining as such, undertaken by Hanson, Australia, who have sand mines both north and south of Perth. The sand mining is being taken place in Banksia woodland, which is the predominant floristic community on the Swan coastal plains around Perth. And across the Hanson sites, there's about 300 or so species that they have to deal with in a restoration context. But the Swan coastal plain and Hanson sites are part of the greater Southwest biodiversity hotspot, which has about 8,300 species there with about 50% endemism. And Perth itself is actually sitting on a part of that biodiversity. And if you look at the map there by Hopper and Goya to your right, you'll see that that orange-red bit is pretty much where Perth is located at the moment. With Hanson's mining operations, once they've completed that, they restore about 7 to 10 hectares of Banksia woodland each year as such. And most of the plants that they actually get to return to their sites from respread topsoil, which is a very important resource for them, which contributes to about 60% of the post-mining species diversity. But as well as getting those species to come up from the topsoil, they also direct seed about 25 species each year as well. Now, the species that they focus on for direct seeding are really things that are bradysporous, so they don't form a persistent soil seed bank. They also have in their mix various dominant keystone species, as well as legumes too, to try to pump some nitrogen back into the system as such. As well, they also use green stock from a couple of species, in particular the eucalypts and Banksia woodland, and their seed resources are actually not handled by themselves. They're actually handled by another company called Trannon Revegetation Systems. Now, Trannon operate a large modern seed bank, which has only been in their new premises for a couple of years now. And the seeds in their seed bank are stored under 5 degrees Celsius and 20% relative humidity, which is a very good standard for storing seeds. And for Hanson, there's about 200 species currently held for restoration purposes. So they have a lot more seeds in their seed bank than they actually use for restoration currently. And this represents about 40 or so different plant families. So they have a big representation of the species diversity found on their sites. And collectively, they're holding about 700 kilograms of seed for Hanson restoration purposes. But beyond Hanson, they hold seed for other activities as well too. And one of the main species they actually have in storage is Nuytsia floribunda. And the Nuytsia in times gone by was used in direct seeding programs, but has proven difficult to return for poorly understood reasons. So what is Nuytsia floribunda? Well, Nuytsia is basically a mistletoe, but unlike most mistletoes that people are aware of, which are epiphytes that are parasitic in tree branches, the Nuytsia floribunda is the world's largest tree mistletoe, if you like. It's actually a root parasite. We've got a picture there of one growing in
correct slide chain.
Michael, are you seeing the the cover slide, or are you seeing the?
We're seeing the cover slide, but I've been flicking through them as you've been going.
Oh, excellent. Okay, thanks for that, Michael. Sorry for sorry for that. Anyway, so it's Nuytsia is a small tree, and it's about one of 15 or so dominant tree species that are found on the Swan coastal plain, but as well as being found on the Swan coastal plain, it's also found right throughout the southwest of West Australia as well. And a region that has about 85 to 90% clearance, unfortunately, mainly due to agricultural development as such. So from a Hansen's perspective, it's actually very common in its pre-mined environments. But following restoration, its return is very poor. And when I've delved into the literature to find out more about what's actually known about it, there's actually surprisingly little information out there as such. Various sources that I've discovered suggest that the seeds are short-lived and they don't store very well. They take a long time to germinate, in some cases up to 90 days or so to germinate. They also have large embryos. There's a report saying that they require boiling to germinate, which would suggest that it has physical seed dormancy. And there's other reports suggesting they might have physiological dormancy as well too. Interestingly enough, that the mistletoes in general have recalcitrant seeds, i.e. desiccation sensitive seeds, which are dispersed within berries. So it does beg the question if one of the problems that people are encountered is to do whether the seeds are actually recalcitrant or not for the Nuytsia. Unusually, Nuytsias actually produce a samara, which is a dry-winged, one-seeded indehiscent fruit rather than a berry as such. So that's another attribute that makes them a little different from the rest of the mistletoes. And these samaras are actually wind dispersed rather than relying on an animal as such. Something seems to be happening. Okay, Michael, can you go to the next slide? Okay, so first, so the questions are really to do with the fact that Trannon has extensive holdings of Nuytsia going back 10 or so years. And it's, as I mentioned before, it's probably one of the most common species in the Hansen seed bank currently. So we set out to answer a series of questions as such. And the questions are, keep on going, Michael. Does Nuytsia seeds firstly have desiccation sensitive recalcitrant seeds? What is the seed quality like across the different Nuytsia age classes? Are Nuytsia seeds water impermeable? What incubation temperatures promote Nuytsia seed germination? How rapidly do Nuytsia seeds germinate? And do Nuytsia seeds have an underdeveloped embryo? And can we treat the Nuytsia seeds in any way to actually improve both the germination rate and seedling emergence as such? And to answer these questions, we actually had access to 4 different Nuytsia age classes going back to 2016. And the most recent age class we had to work with was in 2020. So it was only a couple of months older when we got our hands on it. So what did we find? For starters, when we looked at basic seed attributes, Nuytsia seeds, or the samara themselves, are actually very large. And when you compare it to a database that was done by Angela Moles back in 2005, they certainly are large by world standards as such. We also found that the accessions had very high purity. And purity ranged from about 88% up to 99%. So the seed batches themselves that we were working with were all of very good quality. We also detected little variation in actual seed mass across the different accessions as well. And all the accessions we were working with had a very low seed moisture content, unsurprisingly, given that they'd been stored at 20% relative humidity. We also found that the fill was high to very high across all the accessions, and the seeds, when we chopped them up, all appeared to be white and healthy. So in terms of understanding the basic attributes of the species, they were ticking all the boxes. There was no obvious problems in terms of seed quality, viability, mass, etc. The first thing that we did after looking at basic seed attributes was look at imbibition, just to see whether the seeds could imbibe water or not. And what we found is that they actually imbibe water really, really quickly, and they imbibe water to a very high percent as well. And when you compare them to other species, they're taking up more water than anything with a comparable mass as such. So clearly ruling out water impermeability as a factor, perhaps limiting germination in the species. The next thing we looked at was germination attributes. And what we found is that we generally got quite good germination across all the different accessions. In fact, our oldest accession proved to be probably the best for germination as such. They also displayed quite a narrow window for germination. Optimal germination temperature was between 15 and 20 degrees. 25 degrees proved to be very poor for supporting germination. 10 degrees wasn't too bad, but it certainly wasn't as good as 10 or 15. They also had a T10, which is basically the time to 10% seed germination of about 18 days at least. And you can see from the table here that the optimal temperature for enhancing germination speed was about 20 degrees Celsius. Temperatures higher or lower than that slowed down the germination process quite significantly. But what does 18 days mean? Well, when we compare that to a database of 48 other Southwest WA species, looking at their T10s as such, you can see that Nuytsia, by comparing against them, was very slow by that standard. So technically the seeds are non-dormant, but they are very slow to germinate. The next thing we wanted to look at was embryo morphology. And what we discovered was that the seeds had a linear underdeveloped embryo. As you can see from this image here. So the embryos are about 3.3 mils in length, but they actually grow within the seed prior to radical emergence. And you can see that in the next image as such. So the seeds go from about 3.3 mils in length to about 4.6 mils in length. And it takes about 18 to 20 days for that to happen. And the E to S ratio, which is the embryo to seed ratio, also changes over that time. It goes from about 0.5 up to 0.7 as such. And when we key this out, looking at germination rate, the type of embryo, we've discovered that the seeds actually have morphological dormancy. But when we compare them to species with a similar attribute, i.e. species with underdeveloped embryos that either have morphological dormancy or morphophysiological dormancy, which generally are slow to germinate anyway, the Nuytsia seeds are still slow when you compare against species with a similar attribute. So they're slow regardless of how you look at it. To speed things up and improve seed handling, we looked at acid digestion as such. And basically we looked at sulfuric acid, which has been shown to increase germination speed and overcome physiological dormancy in various species that are in the literature. And it's certainly been proven to be effective for species with indehiscent florets, certainly in grasses as such. And it's been particularly effective for removing hairs and other covering layers and reducing volume and mass in florets as such. So you can see from the image here with the Petrophily linearis, this is a indehiscent fruit with all the hairs and everything on it before treatment, and after being exposed to acid for 2 minutes only, you can see all those hairs have been dissolved away. Typically for sulfuric acid treatment, we look at 1 to 90 minutes exposure, which is all obviously species specific. And we looked at the sulfuric acid in regards to the Nuytsia just to see what impact it had on germination and whether it changed some of the attributes of the samaras as such. We did this under laboratory conditions as well as soil conditions as part of a punnet trial. And we looked at durations between 5 minutes and 30 minutes for acid exposure. And we compared that to the samaras that have been manually clipped as well as intact samaras too. So lab results. What we found was that there's about 80% reduction in volume of the samaras when we actually digested them in acid. So the blue bars here represent the volume taken up by 22 samaras before treatment, and the orange bars represents the same samaras after treatment as such. So you can see there's a massive reduction in volume, and by and large, there was no duration effect. So 5 minutes in acid resulted in a similar sort of reduction in volume as 30 minutes in acid, which was similar to the manually clipped samaras as well. In terms of seed mass, again, we looked at samaras before the acid treatment and looked at the mass of them after the treatment. Again, after treatment, you can see for the acid treated samaras, generally the mass was quite similar after the treatment as such, and the clipped samaras had a marginally significant mass relative to the other treatments that we assess. In terms of germination, what we found was that the samaras that were exposed to acid for 5 to 15 minutes germinated in a very similar way to our manually clipped samaras. It was only the 30 minutes in acid that really reduced germination to some degree. And the images just below there are basically just showing you what the samaras look like after they've been exposed to acid for 5 to 30 minutes. Then when we went to the soil, we only took a subset of the samaras. We just looked at germination on top of the soil as well as in the soil for intact samaras, and then for samaras that had been manually clipped, and then samaras that had been exposed to acid for 5 minutes. And when we went to soil, emergence generally is much lower in sand, unsurprisingly, which we see quite a bit when you go from a petri dish germination trial to a soil germination trial as such. We also found that the samaras that have been treated with acid for 5 minutes generally had slightly lower germination than the other treatments as such. And when we pulled our germination across the 3 main treatments, looking at intact samaras versus clipped samaras versus acid treated samaras, the intact samaras and the manually clipped samaras germinated in a very similar way. It was only the acid-treated samaras that had a reduction in germination or emergence from soil. And likewise, when we combined all our surface sown samara data with the ones that have been sown 1 centimetre below the soil surface, the surface sown samaras generally perform better than the ones that were sown in soil. And in terms of germination speed, the samaras that were on the soil surface understandably germinated more rapidly compared to ones that were buried in the soil. And there's about a 10 to 15 day delay for the ones that were buried under the soil. So the next steps in this work, basically we set up several weeks ago now a number of field trials, and we're going to be looking at how the seeds samaras perform under field conditions. And we're going to be looking at acid treated samaras versus manually clipped ones. And we're just going to see how they go when left to their own devices. We're also looking at intact samaras and see when they germinate naturally under field conditions, and looking at the soil conditions that occur during the germination process as such. And we're also looking at surface sown samaras versus ones that have been incorporated in the soil too, to see whether the results that we observe under field conditions are similar to what we saw in our punnet trial. So implications for industry. Nuytsias appear to be tolerant to current storage conditions, so they certainly don't have, as best as I can see, any signs of being recalcitrant, but they are unusually slow to germinate, which may explain some of the variable results people have reported in terms of their use and restoration. They also have a narrow temperature window for supporting the germination process, which may be another factor as to why they can be problematic in restoration. And they have an underdeveloped embryo, thus morphological dormancy. We certainly found that the acid-treated samaras significantly reduced bulk, and it may be a way to improve storability of samaras, but we might revisit that and cut back on the duration of exposure to acids. So rather than using 5 minutes, we might trial 1 or 2 minutes just to see whether that gives a similar product, but also has perhaps a less of a deleterious effect on germination from soil. And interestingly enough, surface sown samaras seem to perform better than those sown below the soil surface, which is a little unusual. Most of the seeds that I've worked with tend to perform better when you actually get them into the soil, but the seeds of Nuytsia seem to be doing something opposite as such, which I think is quite interesting. And it does beg the question, perhaps, given some of the fussiness in regards to the species, it might be better to actually produce Nuytsia tube stock in a nursery environment rather than direct sowing seeds in the restoration sites, and using the tube stock for later planting when the plants have started to grow well in pots. And that's the end of my presentation. Thank you very much for listening, and my apology for some of the technical glitches.
Thanks, Shane.